Hey guys, so this is the video for Chapter 8. It's the very first one on energy and matter, part of Chapter 8's microbial metabolism. It starts about the diversity of microbes in this chapter. We're going to go over that, but let's get through the first section of what it means for metabolism in the first place. Our case study for Chapter 8, we have Hannah. She's 15 and she's been hanging out with her parents in, where's she at? Gambia. So she's in Gambia, and her parents are working for a non-governmental organization. I don't know, Doctors Without Borders or something like that. So she's down in Gambia working there, helping with her parents working there. And it's been about 3 weeks, and she's starting to show symptoms that she's not feeling well. So she's not hungry, and she is sluggish, fatigued, confused. She doesn't like to be outside, um, especially during the daytime. She's starting to get nauseous. She's vomiting, and she thinks it's the parents think it's a 24-hour bug, but she's not getting better. So I would definitely be concerned if I were her parents. And reflexes are starting to seem slow and light sensitive. So and also stick neck. So she's got some concerning symptoms that need to be checked out. So we got to figure out what's going on with Hannah. We'll check back in with her. As far as metabolism, let's get started. Metabolism is going to be the sum of both of these two types of reactions. So metabolism is everything that's going on in your body or a microbe's body, and it's catabolism or anabolism. So catabolic catabolism, um, some of these concepts in this entire section, they gloss over these things, assuming that you've had these in previous science classes. Um, so I know some of you have and some of you hadn't. I'm going to try and do my best to cover them without going into too much detail, but still getting the explanation in there. But I want y'all to know these things. These are basic concepts. If you get these down, then the rest of the chapter should make more sense. So we should have covered catabolism and anabolism in a previous biology, or at least anatomy and physiology, before you took this one. If you took that one, or bio 1010, hopefully you've covered this stuff, or even in chemistry. But let's get through it. Catabolism is going to be where you take a big molecule and you break it down. So our favorite molecule that we talk about in metabolism is glucose. And so you break down glucose, right? All of us are breaking down glucose for our energy source. And at the end of breaking down glucose, you're going to end up with smaller molecules. So glucose is a big sugar, sort of. It's a bigger molecule than carbon dioxide. So glucose, C6H12O6, we're going to break it down into carbon dioxide, CO2, that we're going to exhale. And then we're going to break it down. It also breaks down into water, which is also a smaller molecule, H2O. So in catabolism, you make a big molecule and you break it into smaller molecules. When you break those chemical bonds, you are going to release energy. When you release energy, it's also called exergonic, because energy is releasing from the system. Energy is being released, and hopefully you can harvest the energy in form of ATP. We'll get to that in a little bit. So to summarize, catabolism, you take a big molecule, you bust it up, you break the bonds, and when you break the bonds, you release energy. That big molecule is broken down into smaller molecules. So that's catabolism. On the other end, the other side of the coin is anabolism. Anabolism is going to make a big molecule out of smaller molecules. So with anabolism, we are going to take small molecule, small molecule, small molecule, small molecule, and we want to make it into a bigger one. But we can't do that without energy coming into the reaction. So we've got to add energy into the reaction to build a big molecule out of little molecules. So anabolism takes energy into the system. So this is going to make it endergonic. Energy is coming inside, endergonic inside. If it was catabolism, energy is exiting because you broke the bonds, and it is exiting exergonic. Metabolism is going to be the sum of both of these. Another way to memorize these, anabolic, if you think of people, guys in particular, that possibly take anabolic steroids. Of course, there are ladies that do this too. But people that are bodybuilders trying to bulk up, anabolic steroids, trying to bulk up and make their muscles bigger than they were before. So anabolic is going to synthesize a bigger molecule from smaller molecules. Catabolics are going to take a big molecule and break it down. So I always say, I've got cats in my house. If you didn't see me in the previous video, I have 2 cats, and what they like to do is shred things. They love to shred things. So they will shred my couch into teeny tiny little fabric strips. Catabolism breaks things down into small things. Anabolism builds things up from small things into bigger things. All of these together is metabolism. Metabolism is the sum of all this stuff. So speedy metabolism, lots of catabolic and anabolic going on. If you got a slower metabolism, somebody is Hashimoto's disease. Their thyroid's going really slow. They've got a slower metabolism. These reactions aren't happening as fast as somebody with a faster metabolism. Okay, spent way too much time on this concept, but wanted to cover those basics. Examples of this. Here is what I was telling you about, glucose for cellular respiration. We're going to go in that into section 8.3. But we got glucose. We are aerobic when we undergo cellular respiration. Steps 2 and 3 out of cellular respiration require oxygen. So we take in a molecule of glucose and 6 molecules of oxygen, and at the end we get smaller molecules, 6 molecules of water and 6 molecules of CO2, carbon dioxide, that we're going to exhale out of our lungs. And then we broke the bonds, we released energy, and we're going to capture that energy in one of our favorite energy currency molecules, which is ATP. So this is glucose broken down with cellular respiration. This is a type of catabolic reaction, and we're going to harvest that energy in the form of ATP. Other example, which is basically just flipping the arrow in reverse, is photosynthesis. And photosynthesis is happening with plants are photosynthesizers. There are other types of cyanobacteria that are photosynthesizers. So they're going to take the CO2 that you and I are exhaling and putting in the atmosphere. They're going to take water, and they're going to take those 2 smaller molecules, use the energy from the sun. So energy is going to be the transfer of electrons, whether those electrons are coming from light energy and photons, or if that energy is coming from chemical energy. But they're going to take a small molecule, and the energy, and they're going to build up glucose, which is a bigger molecule. And then the byproduct is oxygen. So to take small things and build a bigger thing, this is an anabolic reaction. Here again are those concepts of exergonic and endergonic. Energy is exiting because we broke bonds in an exergonic reaction. Energy is entering when we need energy to build a bigger molecule out of smaller things. So cellular respiration, breaking down glucose, and it breaks down into these molecules. This is a catabolic reaction, and energy is going to be exiting. Our energy molecule is ATP. That's our energy currency molecule. When we break the bonds of ATP, that energy is going to go out, and that energy releasing from ATP going down to ADP is going to release that energy, and we're going to use that to power other things throughout our body, like sodium potassium pumps or something that needs energy. Lots of things need energy. ATPs are energy currency. We're going to snap off phosphate there, break that bond. We break the bond, releases the energy. And then later on, we've got ADP. When we bring in energy, we're going to eat dinner or lunch or breakfast, and we're going to bring in, for us, chemical energy. That energy is going to tack on that third phosphate back on to ADP, adenosine diphosphate, and now it will become ATP. So ATP is our energy currency molecule. It is a rechargeable battery. We snap off the third phosphate, break that bond, we release energy, and we can use it throughout our body for lots of things. Then we're going to bring in energy in the form of, for us, we're chemotrophs, we're chemoheterotrophs. So we're going to bring in energy. That energy is going to be used to transform ADP and tack on that third phosphate back again to make it into ATP. If energy is releasing, exergonic. If energy is coming in, endergonic. When we classify organisms, we can classify organisms by energy and then their carbon source. If we're classifying them by energy, are they getting their energy from light? It's a phototroph. If they're getting their energy from chemicals, it's a chemotroph. So that's 2 methods of classification right there. What is their energy source? Now, then we break it down. what is their carbon source? If their carbon source is going to be inorganic, then these guys are going to be photoautotrophs. If their carbon source is an organic, then they are photo because they still use light as an original energy source. And then they're going to use organic carbon and they are photoheterotrophs. So plants, that process that I talked about with photosynthesis, plants are going to be photoautotrophs. Plants, algae that we talked about before in previous chapters, golden algae, red algae, green algae, or cyanobacteria, all these guys are photoautotrophs. They are going to break down inorganic, they're going to use energy, light energy, and then they are going to use inorganic carbon, CO2, versus organic stuff. Chemotrophs are not going to use light, so there's no photo in there at all. It is a chemotroph. Now, what carbon source are we going to use? If we're inorganic or are we organic? We are chemoheterotrophs because our carbon sources are organic compounds. We talked about the macromolecules before: carbohydrates, lipids, proteins. All of those are energy sources for us. So we use organic compounds. We are chemoheterotrophs. So all animals, chemoheterotrophs, as well as fungi and some of the protists and some bacteria are chemoheterotrophs. I believe the textbook said that, what did it say? I think it said E. coli was a chemoheterotroph. Find it. Oh, it's in here somewhere. Yes, E. coli. I was right. Okay, so chemoautotrophs, again, if it's inorganic carbon, it could be CO2. And those guys, they could use CO2 or they may also use something else. So another table that I like to use besides this boring one here from the textbook, I like this table that I put in the PowerPoints because this breaks it down by energy source, just like that other table. Is it chemical energy source or is it light? If it's light, it's photo. If it's chemical, it's chemo. And then out of your chemotrophs, are you going to have organic compounds? And if you are, it even breaks it down into the final electron receptor. But again, I said the animals, fungi, protists, and bacteria are all going to be chemoheterotrophs. Chemoautotrophs, they're going to use CO2 as their inorganic carbon source. And then their electron receptors, they may break down and reduce other compounds like hydrogen sulfide or iron or something like that. And so these guys are lithotrophs. They are chemoautotrophs. They don't use light at all. They're going to use chemicals as their energy source as well as their carbon source. Over here on the phototrophs, are they going to use organic compounds, photoheterotrophs? Are they going to use inorganic, CO2, photoautotrophs? And out of our photoautotrophs, we were talking about plants and algae and the cyanobacteria, common photoautotrophs. But there are also other guys, our photoheterotrophs. So our green and purple bacteria that we mentioned before, those guys are photo because they use light as the energy source, but then they're going to use organic compounds and break them down as their carbon source. These are photoheterotrophs. So I like this diagram a little better simply because it gives you these examples in these yellow boxes of the different types. So I like this picture a little bit better than this table from the textbook. Okay, so we said energy is coming in the form of electron transfer. And those electrons could come from light energy, from a photon, or those electrons could come from chemical energy. So when we talk about the electron transfer, there's two things. You're either going to lose electrons. If you lose electrons, if you're the molecule, if you're the substance that loses electrons, it's called oxidation. If you are the substance that gains the electrons, if you are gaining electrons, it's called reduction. So reduction is going to be gain of electrons. Oxidation is loss of electrons. When you have a chemical equation, these are both happening at the same time. Just like we said, catabolic and anabolic, and both of those summarize to metabolism. Oxidation is happening and reduction is happening on either side of the equation. So we've got magnesium and we've got oxygen here in the picture. If something gets oxidized, so we've lost electrons and we moved it over here, we lost these two guys over here and we moved them. Oh no. Uh, and then over here, oxygen, here's our oxygen molecule. It is going to gain electrons. So oxygen has gained them over here. So if we have oxidation going on, reduction going on, it is called a redox reaction. Do you understand the phrase oil rig if you haven't heard it before? Oxidation is loss, oil, reduction is gain, oil rig. Know that. It will help you understand who's gaining electrons, who's losing electrons, what term are we talking about? Are we talking about oxidation or are we talking about reduction? So know that phrase. I'm sure it's going to be on the test. This common concept, talking about moving electrons, who gets them, who's losing them. Energy carriers. Who's moving around the electrons? There are molecules whose job is to transport electrons. There are great molecules that do this. ATP is one of them, but there is also NAD, NADP, and FAD. And so we can see NAD plus is on this side. And on the other side of the arrow, we have NADH. So hydrogen got tacked on. Hydrogen is going to bring over all of its subatomic particles. It's going to bring over its protons. It's going to bring over its neutrons, and it's going to bring over its electrons. So if we didn't have a hydrogen before, and now we gained a hydrogen with its electrons, we've gained electrons. What did we call that? Was that oxidation or reduction? If we remember the phrase oil rig, we remember reduction is gain. So this is reduced. We went from NAD to NADH. We gained electrons. We were reduced. So you will see these molecules have switch. They'll gain and they'll lose electrons constantly throughout these things. So energy carriers, NAD, NADP, FADs, ATPs are energy carriers. They can shuttle electrons back and forth. Here is a better picture of an ATP that I was telling you was our energy carrier molecule. It's got the 3 phosphates. Adenosine triphosphate is what it stands for. Adenosine, we've got adenine, our nitrogenous base. Here's ribose is our sugar backbone, just like our nucleotide for DNA that we're going to get into later. And then we've got 1, 2, 3 phosphates. So it is triphosphate, the TP out of the ATP. So ATP is 3 phosphates. When we snap off that 3rd phosphate, I told y'all we break a bond. And when we break bond, energy gets released and we can use that energy to power things in the body. So when we snap off that 3rd phosphate, we are now adenosine diphosphate because we only have 2 phosphates on there. And that 3rd lone little phosphate, sometimes you'll see them little PI, is how they designate it. And so it's adenosine diphosphate and the 3rd phosphate. We're going to need energy to come into the system, whether it is solar energy or chemical energy. We could be photo or we could be chemo. But some kind of energy is going to be needed to tack on and go from ADP to ATP, tack on that 3rd phosphate. So it's got to be an inorganic reaction. Energy has to come in. And it's going to synthesize a bigger molecule from ADP with 2 phosphates to ATP with 3 phosphates. So ATP is our energy currency. And when we go into the cellular respiration section in 8.3, we're going to count up how many ATP we get at each step. Activation energy. So to get from point A to point B on a chemical equation, we are going to have activation energy. If you had a substance, it's not just going to break down on its own. So if we had glucose, if we had sugar just sitting in a bowl in your packet that you're waiting to put into your coffee, it's not just going to break down into CO2 and water. It just doesn't do that. It's got to have some sort of catalyst to start the ball rolling, to start the process. So some of these things are enzymes. Enzymes are great catalysts, and a lot of our enzymes are made out of proteins. And they are going to help lower the energy needed to start the process, to start the ball rolling. So here we've got a reaction going on, and it's in green, and it doesn't have a catalyst. So it is trying to perform this reaction. Say we're trying to break down lactose. So we took in some cow's milk, and it's loaded with lactose, and we don't have lactase. So now we are going to end up using a whole lot more energy, a whole lot more activation energy without the lactase than if we had the enzyme lactase. If we have that enzyme, it's going to lower the activation energy to be able to break it down, to be able to break down lactose into the smaller, simple sugars. So activation energy is what we need to start the ball, to start the reaction going. If you have an enzyme, it lowers the amount of energy needed to start the reaction, to get the reaction going. So lactase is an enzyme that helps break down lactose. Catalase is an enzyme that's going to break down hydrogen peroxide. And so in our face-to-face labs, which we didn't get to do, but at the end of our face-to-face labs, we talk about one of our lab modules is trying to do a test to differentiate, am I looking at streptococcus or am I looking at staphylococcus? So we have a sample of Staph aureus and we have a sample of strep, and we're going to put them on slides, and then you put peroxide on them. And what happens when we put peroxide on our body? We have catalase, right? So catalase breaks down hydrogen peroxide and it breaks it down into water, and then those oxygen bubbles that bubble up on our skin if we put peroxide on our wound. So we have catalase that breaks down hydrogen peroxide. When you do this test, differentiating between Staph aureus or strep, Staph aureus has catalase, and if you put peroxide on it, it will bubble like it does for us. If you put peroxide on the strep, it doesn't bubble, it just stays liquid, and it doesn't break down because it doesn't have catalase. So lactase, catalase are all examples of different types of enzymes that help lower activation energy to break down a substance with less effort, basically. All right, so we were talking about enzymes and how they lowered the activation energy needed for a reaction to occur. So how are they doing this? What are enzymes doing? So here we've got an enzyme, and we mentioned, I'm going to go with the example that we talked about before. So we mentioned catalase, and we said that it breaks down hydrogen peroxide. That's going to be our substrate that gets broken down by our enzyme. So we said enzymes, they're proteins, and protein shape matters. We said that there's primary, secondary, tertiary, and quaternary structure of proteins. So the way that they are built and the shape that they have truly matters. So that shape that they have, they're going to have some place on there where this substrate, in our case, peroxide, the substrate is going to bind. So it has to bind, and that substrate has to match this location. So it's a lock and a key, where our enzyme is the key, our enzyme is the lock, sorry, and the substrate is the key. When they meet at that place where they come together, that place has to match, and so it is called the active site. The active site. So the substrate binds onto the enzyme at the active site. When they bind, now we've got an enzyme substrate complex, and then the magic can happen. So catalase is going to bind with the peroxide, and what happens when catalase hits peroxide? It's going to create the products, and it is going to end up with the two products that we talked about. We're going to have water left behind, and then you're going to have the oxygen molecules that bubble off as oxygen gas. So if you've got catalase and peroxide attaches to, binds to at the active site, we are going to get the products produced. We're going to have water left behind and oxygen gas that bubbles away. So it's called the induced fit model because when these two bind, when catalase binds with the substrate, it is going to change the shape of both of them just a little bit. And so it's called the induced fit model when this happens. It's basically a lock and a key though. The key fits at the active site to bind into the lock. And then at the end, we're going to produce some kind of products That is what we hope to do with an enzyme. So it's going to lower the activation energy and it's going to create the products that we need to happen. Sometimes it can't do this alone. It's got to have its best friend, its ride or die, and its best friend could be a cofactor or a coenzyme, some kind of vitamin or mineral, something comes along that binds here. And now, when this cofactor binds, we've got the proper shape at the active site. Because before, this is our active site shape, and that isn't what we're looking for. That's not the right shape. We got to have our buddy. We got to have our cofactor. We got to have our coenzyme. Maybe it's CoQ. There's a whole bunch of different vitamins out there that are great. Coenzymes, cofactors, whatever we need that will be our buddy for the enzyme to create the proper shape of an active site. So here we're going to have our cofactor bind. When we don't have it, when it's solo all by itself, it's called an ABO because A means without. It's an enzyme without its cofactor. When we do have our best friend, we're whole, we're made whole. So we're a holo enzyme. So now we've got our enzyme and our cofactor, our coenzyme, and we've got the proper active site that can bind and match up with our substrate. And now we can continue. So if the enzyme can't do it solo and needs a best friend, the best friend is called a cofactor or a coenzyme, and together they make the great shape of an active site for the substrate that they are bound to attach with. When we talk about these reactions, we are going to inhibit, when something's inhibited, it's blocked, or we can activate and turn it on. So if something's inhibited, it's not producing what it needs to produce. How can that happen? Why does something get inhibited? Well, we got two ways for inhibition to happen. So we said that here's our enzyme, and our enzyme had this place to bind called the active site. With competitive inhibition, a competitor comes along. So this jerk comes along and it takes up the active site. So our competitor, it's going to physically block the active site. It's stuck in there and put itself in there and block the active site. And now our substrate can't come along and it can't bind. So the substrate's left high and dry. It can't bind. We're not going to produce what we need to. It's inhibited. And it's inhibited because the competitor came along and blocked the active site. What a jerk. There's another type of inhibition too. It is called non-competitive inhibition. It's also called allosteric inhibition. And it's called allosteric inhibition because it's binding at another place. It's not the active site. It's called an allosteric site. And that can be anywhere else besides the active site is an allosteric site. So this thing comes along and binds someplace else. When it binds someplace else, it's called an allosteric location, an allosteric site. So now we have a competitor and it's binding in a different location. But we said again, protein structure matters. The way it looks, the way it's shaped matters. And since this guy came along and bound at the allosteric site, we lost our happy smiley face looking Pac-Man shaped active site. And now Pac-Man's mouth is closed and he cannot bind to our substrate, and our substrate still is left high and dry and cannot bind. So in both of these are both types of inhibition. They are inhibition, competitive inhibition, if it attaches at the active site location. It is non-competitive inhibition or allosteric inhibition if it binds at an allosteric site and changes the shape of the active site. It's not like this, it looks like this. And both of these result in the active site doesn't look the way it should, and the substrate has to go on its way alone, solo, no products. This is inhibition. Activation, so we talked about allosteric inhibition was non-competitive inhibition. It means the same thing, just different terms, same thing. There is allosteric activation. So again, if something binds at a place other than the active site, it's an allosteric site. So here we have something come along and it's an activator. It's going to bind to the active site and, sorry, not the active site. It binds to the allosteric site. Before, we had this weird spot and it didn't look like the proper active site. The proper active site for this substrate looks like Pac-Man with his open mouth. So we didn't get that structure up here because we didn't have the activator come along and bind. Once the activator came along and bound at the allosteric site, instead of blocking it, instead of inhibiting it and changing its shape for the worse, this actually changes its shape for the better. So allosteric activation changes it for the better, and now we have our happy Pac-Man shape for the active site, and it is able to gobble up and bind to the substrate, and we end up producing the products that we need to. So there's allosteric inhibition, where we change the active site and it doesn't work. There's allosteric activation, where we change the active site and it does work. And then the last thing having to do with these enzymes is feedback, and it's called inhibition because, again, inhibition stops a process. It's going to shut it down. So this is just like those hormone cascades. If you've made it through AP2 and we talked about feedback, and there's negative feedback and positive feedback, and almost everything was negative feedback. We talked about in the hormone chapter, chapter 17 in AP2, we talked about the hormone cascade that can happen. So hypothalamus is going to release a hormone that then goes to your pituitary gland, and the pituitary gland releases a hormone that goes to the bloodstream, and it ends up with the gland that it hits up, and that could be your thyroid gland. Then the thyroid gland is going to release the hormone T3 and T4. That's going to go into your system. It's going to speed up the metabolism, and then when the T3 and T4 gets high enough, that's going to get picked up by your hypothalamus, and your hypothalamus is going to go, hold up, we're good, and shut down the whole process. So that is called feedback inhibition. So here is our original substrate coming, for the example, from the hypothalamus, and then it's going to go and hit up from the hypothalamus. It's going to hit the pituitary is then going to produce this guy. This guy is going to hit up with the thyroid gland is going to produce T3, T4. That's going to produce something else, and then at the end product is going to eventually shut down the beginning product. So it is negative feedback inhibition. So that's the last of this first section in chapter 8.